welcome Dr. Narasimha Murthy for his talk. Dr. Murthy is the founder, director of Institute for Drug Delivery and Biomedical Research based in Bangalore. He's a renowned scientist in the field of topical drug delivery. He has received several international awards for both his scientific research and philanthropic activities. He will be talking about drug delivery systems, a pragmatic approach to enhance the efficacy of therapeutic agents today. Welcome, Dr.
So good afternoon, everybody. My name is Narasimha Murthy. I'm the founder director of Institute for Drug Delivery and Biomedical Research. It is a non-profit research organization. The title of my talk today is Drug Delivery Systems, a pragmatic approach to improve the treatment outcome. Just a brief introduction to my organization before I proceed with the presentation today. This is the facility, Institute for Drug Delivery Biomedical Research. It is located in Bangalore. Basically, it's a non-profit research organization. It was founded in 2013 to foster research in the space of drug delivery systems to find solutions to unmet medical need. We've been around almost close to 8 to 9 years. Most of our objectives and goals are in line with Dr. Sridhar just described here, so I don't want to again reiterate the same thing. We do a lot of philanthropic activities. We support the academic institutions. We have students coming in as interns. We help them write, publish papers. So a lot of activities almost on the same lines. So in this organization, we have a bunch of experts who work in different areas of drug delivery systems. We have people who work on targeted drug delivery, like similar to exosome technology, not exactly exosome. It's other nanoparticulate and liposomal drug delivery technology. We have people who work on dermal transdermal like me, and people who are working on oral drug delivery systems and several other areas of research. So basically, a drug delivery systems, as you all know, is a broad term, right? So we are referring to something where we are trying to deliver the drug through different routes in different modes. Okay, so basically, drug delivery systems are intended to deliver drug to the target site in therapeutically relevant modes. So the drug delivery systems is a great approach to reduce the dose of the medication. Like just now we had a talk from Dr. Reddy's. He showed how the medication dose was reduced from 100 milligrams to 50 milligrams. That's a great improvement in terms of drug delivery. And then it can also, if you choose an appropriate drug delivery system, you can reduce the cost of therapy significantly. And there are so many drug delivery systems like sustained release and controlled release delivery systems that would prolong the therapeutic level of drug in the systemic circulation and prolong the therapeutic activity as well. So there are some approaches that are meant to improve the compliance of the medication, particularly in sensitive population, like pediatric population, something like taste masking and odor masking approaches. So drug delivery technologies has several responsibilities, several roles to play. Personally, I work on dermal and transdermal delivery systems, so I want to discuss the first few slides on dermal and transdermal delivery, giving you a few examples on how the research, what is the direction of research in this field. So basically, topical and transdermal drug delivery research has been going on since several decades. We know quite a few facts about skin now. Unless the drug molecule is small and has a log P value between 1 and 3 and has a low melting point, it will not be able to make its way through the skin layers. Basically, skin has multiple layers. The topmost layer is the stratum corneum, which is basically filled with keratinocytes, and intercellular space is filled with lipids, which are hard to permeate. And there is a viable epidermal layer, which has all living cells and a lot of water in it. And then, of course, the dermal region where you have the blood vessels and the nerve endings and everything, like a normal tissue. So there is no blood vessel in the stratum corneum and the epidermal region. I'm just reiterating it. There is no blood vessels or nerve endings in the stratum corneum or epidermal region. This is useful for me to explain, the further technology that I'm going to talk about in the future slides. So stratum corneum is poorly permeable to large molecules, or if it's a hydrophilic molecule, the lag time to achieve a steady state diffusion would be longer. So always, we prefer to have a molecule in this range. But as researchers, it is a challenging thing for us. We try to explore means and ways to improve the skin permeability to larger molecules and to hydrophilic molecules, or molecules that are required in higher doses, right? So that's our research arena, basically, right? So there are several simple ways of achieving this, like passive ways of improving the skin permeability by the way of using skin permeability enhancers, or there are so many active ways of enhancing the drug delivery across the skin. People have been exploring the use of electric current, like for example, the technology called as iontophoresis, where you use a small amount of electric current to drive the drug, like charged drug across the skin. And then there is something called as magnetophoresis, where you drive the diamagnetic materials using a magnetic field. And there are other technologies like sonophoresis, where you use ultrasound. Okay, the recent one, the recent promising technology and upcoming technology is microneedle technology. I'm going to talk a few bit about microneedle technology in the future. So just to give you an example of how simple solutions can impact the therapeutic outcome significantly, we did a research recently on delivery of pilocarpine. Pilocarpine is basically a sweat-inducing agent in the skin. Okay? It's used as a sweat-inducing agent in the skin. So why would we want to induce sweating? Because we want to measure the chloride concentration in the skin in babies as a potential diagnostic tool for cystic fibrosis. How is it done right now? They're using iontophoresis technique. Okay, they apply iontophoresis in the clinic for small babies for about half an hour. Okay, so and then deliver pilocarpine, and later, once the pilocarpine goes in, it stimulates induces the sweat glands to produce sweat, and you collect the sweat and measure the chloride concentration. It needs to be done quickly, basically. If you apply a passive formulation, it won't happen so quickly. Okay, because pilocarpine is not all that great drug in terms of skin permeability. Okay, so we have to do something to make pilocarpine penetrate rapidly into skin to avoid the use of this electrical technique, because it's a very cumbersome technique. Not all kids are highly tolerant to the electric current level they apply during iontophoresis. So we came up with an excellent combination of permeation enhancers that facilitated a rapid delivery of pilocarpine into skin. So in fact, the passive delivery was almost on par or better than the iontophoresis technique. We evaluated the workability of this technique in human subjects, in about 20 human subjects, and we found out that just about 30 minutes application of this formulation induces enough sweating to conduct the analysis, chloride analysis. This is another example where we tried to deliver a pain modulator, anandamide, which is an endocannabinoid. It is a great molecule, but the problem with this is it's poorly permeable into skin, and it is also prone for enzymatic degradation by enzymes. So our task was to improve the delivery of anandamide into skin, plus mitigate the effect of enzymatic degradation. So we came up with a natural FAW inhibitor by docking studies and such things, and then this FAW inhibitor was also like poorly permeable. So we developed a formulation incorporated with several permeation enhancers in it, and we built in a technology in it to get enhanced permeation of both the FAW inhibitor as well as the API. So we tested this formulation in thermal hyperalgesia and mechanical allodynia moderate. Compare the green bar with the purple bar. You see that in case of green bar, the therapeutic activity lasted only for up to 3 hours, and then when we incorporated the enzyme inhibitor, we could prolong the therapeutic activity in both the animal models for a significantly longer duration. So moving on to the active technologies, basically I'm going to talk about the microneedle technology. So when you inject the formulations using a hypodermic needle, the hypodermic needle is going to penetrate far deep into the skin and touch the neurons and damage the blood capillaries. So that's why you perceive pain. Okay, what if these needles are smaller. Okay, so if there are micron size, like around 250 to 300 micrometer, and if it's made made in the form of an array and applied on the skin, these microneedles would penetrate only in the top few layers of the skin where there is no nerve ending. So that's why the patient does not perceive any pain, or he would not bleed either. Okay, so with this approach, people did a lot of research using metallic microneedles. The metallic microneedles were coated with macromolecules. The skin, the main barrier for skin permeation of macromolecules is the stratum corneum. So this microneedle technology is perturbing that barrier, right, and delivering the drug molecule right into the skin tissue. So eventually the drug that gets into the epidermis and the dermal region can go into the systemic circulation very quickly. Okay, so with this approach, people started working on polymeric microneedles. So polymeric microneedles basically are prepared or fabricated using dissolving polymers. These are biodegradable, biocompatible polymers. You make microneedle that would just dissipate in the tissue within a few minutes. So you incorporate the drug in these microneedles, which would eventually be released in the skin tissue rapidly. So this is not an irreversible damage done to the skin. You see the physiological water evaporation rate of an intact skin, and this is the microneedle treated skin. Immediately after microneedle treatment, the water evaporation rate increases, and within about 6 to 12 hours, the physiological water evaporation rate recovers. So that means it's not it's a reversible damage done to the skin. It's not really dangerous. And then we used this technology for delivering rapid delivery of nicotine for treating smoking cessation. So recently we started working on something called as polymer-coated polymeric microneedles. So we are coating the microneedles using a semi-permeable water-insoluble polymer so that we can release the drug in a controlled release fashion into the tissue. So these are the electron microscopic pictures of the uncoated and coated microneedles. I deliberately ruptured one to show you the uniform coating. And this is the one that was exposed to the fluid, the biological fluid, over a long period of time. I just cut this into just to show the two layers, the coating and the core layer. And we're also working on microneedles incorporated with adsorbent to add another rate controlling step. Basically, the drug need to dissolve into the core microneedle and then eventually diffuse out. So this would prolong the drug release up to 10 or 12 hours. So that's the target duration that we are focusing on right now. So we are exploring the application of these coated microneedles in ocular delivery, basically to the back of the eye. Otherwise, the only alternative right now to deliver drugs into the back of the eye is intraocular injection, which is not a very popular way of doing it. Okay, and then I'm showing you how the drug was released in a controlled release manner when we used a coated microneedle compared to the uncoated microneedle. So the coated microneedle would be of advantage here because it is not dissipating all the polymer content into the vitreous humor. It is only allowing the drug to diffuse out into the vitreous humor. So we're also exploring the delivery of drug molecules using polymer-coated polymeric microneedles for sublingual delivery of drugs, which are used for treating cardiac disorders like cardiotonics and nitroglycerin and such things, which are used in treating angina pectoris and stuff like that. So this particular project is funded to one of our very young investigators by BIRAC. It's a big grant. So with this, I would like to conclude that drug delivery technology is the way to go forward. Personally, I feel that the next generation therapeutic research is going to focus on rendering smaller molecules and the biologicals more safer and effective by using appropriate drug delivery technologies. So I would like to thank all the team members at IDBR. Thank you very much for your patience.
Thank you, Dr. Murthy, for a really fascinating talk and technology. I can see just one hand there, and maybe the rest of time we can take one question.
Hello. Thank you, sir. It was a really good talk. So a similar design model has come recently from a Swiss or a Finland lab. Along with that, a similar, so one more model exactly like this is working in NIAB as well, where they're trying for drug delivery. So how your system is different from that, and what are the advantages over it?
Similar system, you mean the microneedle technology?
Yes,
Oh,
because this
people are-
microneedle technology is pretty old, I guess.
Right.
So how your delivery system versus theirs would be a little different, or is a little different?
A good question. You know, microneedle technology has been around past several years, right? So this polymer-coated polymeric microneedle is not something that is that's been explored so far. Particularly, the microneedle technology was mainly explored for transdermal, dermal and transdermal delivery so far. Okay, so we are trying for intraocular, particularly the posterior segment of the eye, using the polymer-coated microneedle. So that's a novel technology basically. Ours is the first few papers you would see in the literature.
Thank you very much. It's such an interesting talk to go to a painless delivery of the I was just keen to know, just like when you give these intradermal injections, vaccines, and you get a reflex reaction, is there an immune response to your polymeric?
Well, just as the, just for the plain placebo polymeric microneedles, we did not see any immune response. Basically, the polymers that we use here are something like hyaluronic acid and polyvinyl pyrrolidine, which are all GRAS approved, and they do not induce any kind of immune reactions. So it could be, a response to an ingredient that you incorporate into this, maybe like a protein or an allergen and something like that.
My sincere apology for not taking all the questions, from the students as well as the other members. But, he is available, over the lunch, so yeah, we can continue the discussion.
Sure.
Requesting Professor Abhijit to hand over the memento to Dr. Murthy. Yes, sir. Thank you very much for that very interesting talk. So we are announcing the lunch break now, but just before we all head off to lunch, we'll request for a very quick group photo. So if we can all gather up in the center, please.
बंद करो.